Hi everyone, this is the fourth and final video of Lecture 2, Metabolism and Enzymes. So we learned a lot about enzyme in this lecture and we are not stopping there. So what we need to know now is how to name enzymes. In the past, several years ago, enzymes usually named by their substrate or their activity with the suffix "ase". For example, urease, DNA polymerase, ligase, protease, lipase, and sucrase, for example. But some systems, they name the enzymes by the name of the person who discovered that activity. For example, pepsin, lysozymes, or trypsin. So that's in some specific enzyme, there could be at least, there could be more than one name. For example, protease can be either pepsin, can be trypsin. There are many types of protease, and in one enzyme it can have more than one name. So later, similar to the SI unit, the biochemistry consortium, they decided now they are too much confusing. They, they need to come up with a systematic name, and that's named by their catalytic reaction. And we call the name of the enzymes as the EC number. It stands for enzymes commission number, and it's based on catalytic reaction, and we can classify enzymes into 6 classes: oxidoreductase, a transfer of electron, or sometimes you can see it can be the hydrogen or hydride ion. In the second type is transferase, is the transferring the group, chemical group. Hydrolase is the hydrolysis reaction or the transfer of functional group to the water. Lyase is an addition of group to the double bond or removing a group from double bond. Isomerase is more like transferring a group within molecules to yield isomer. For example, you have a chemical compound with some specific attachment right here, some group, and isomerase will move it from this position to somewhere else, like from to here. This is the same chemical molecules, but just a different structure. Ligase is the formation of carbon-carbon bond, carbon and sulfur, carbon and oxygen, and carbon and nitrogen, by condensing the reaction coupled with the ATP cleavage. Alright, this is an example. I try to make my pointer. Alright, here. So this is example of one reaction. You have ATP and you have the glucose as the substrate or reactant, and then you get ADP as a product together with the glucose 6-phosphate. For this reaction, the enzyme that catalyzes this reaction is called ATP glucose phosphotransferase. From this name, you can see that there will be a transfer of a phosphate group, probably from ATP to glucose, indicating the function, the substrate, and the products of the reaction. For the EC number is 2711. 2 is means the class of the reaction, which is transferase. 7 is the subclass phosphotransferase. The first one is phosphotransferase with a hydroxyl group as an acceptor, and one is the D-glucose as a phosphoryl group acceptor. The common name is hexokinase. You can see that just one enzyme, you have at least 3 names, but the EC number is just specific that it need to be this specific enzyme. It cannot be otherwise. And there are a lot more names of the enzyme, and you can go to this website to see the list of that if you want to. I don't actually. Anyway, in our lab, we have 3 examples of enzyme reaction that you can play around. The first one is dehydrogenase or oxidoreductase. Is the transfer of an electron. Alpha amylase is enzyme in the group of hydrolase is breaking the glycosidic bond, the bond between the glucose molecules in amylose, which is a starch. And the last one is the sucrase, or the other name is invertase. It's the hydrolysis of glycosidic bond between fructose and glucose in the sucrose molecules. So let's go to the first reaction. First enzyme, dehydrogenase or oxidoreductase. Oxidoreductase is actually coming from oxidation and reduction reaction. That means it's a transfer of electron from the donor to an acceptor. And it's required coenzymes to carry the electron. That coenzyme in the biological reaction, you will see a lot in the following lectures: NAD+, NADP+, FMN, and FAD. This is the example of the reaction. NAD+ or NADP+ will receive 2 electrons with 2 protons. Together, they form NADPH or NADH with a proton. There are more than 200 enzymes that are known to catalyze the reaction in which NAD+ and NADH are accepting an hydride ion from the substrate that has more of electron. We can call it reduced substrate. The substrate that has a lot of electron, and donate that electron, donate the hydrogen with an extra electron. We call it hydride ion or H minus to another substrate that need an electron. We call it oxidized substrate. Here you have hydride ion or the reduced substrate A with hydrogen, and you have the oxidized substrate. You have this one, NAD plus to accept the hydride ion, and you will get just an A and NADH on proton, similarly to NADPH. Another example is alcohol dehydrogenase that are in your liver to process alcohol into acetaldehyde when you drink. So from ethanol, you have the NAD+. Ethanol have extra hydride ion right here, and it will combine with NAD+ to form NADH and acetaldehyde and proton. These are examples of dehydrogenase. The next one is alpha amylase. For alpha amylase is an enzyme that breaks the glycosidic bond between glucose molecules. This is the structure of starch or amylose or amylopectin. Alpha amylase will break this bond. It can be just right here at the end, in the middle, whatever, wherever it finds the glycosidic bond, no matter where the bond are. And alpha amylase is hydrolase, so it needs water in the reaction. The products can be a big chunk or small chunk or just 2 or 1 molecules of glucose. So we can detect the reaction by observing the the changes or the disappearance of the substrate or the production of the products. In this case, for detecting alpha amylase activity, you detecting the disappearance of the substrate. The substrate in this example is the amylose. This is the strand of amylose is from a coil structure over an iodine. This is in iodine solution, it will form a rod of iodine. And the amylose will form this structure. If you take a look at the top view, you will see the amylose strand and the iodine rod in the middle. And this structure will absorb the light and you will see it in dark blue color. When the amylase works, it will chop up all the strand of amylose and they can no longer form this structure and you will not see the dark blue color anymore. Finally, the enzyme that you have a chance to take a look in the lab is sucrase, or we call it invertase. You can use either name. In this reaction, it is the breakdown of sucrose. Sucrose is the sugar with 2 molecules of sugar. Sucrose is a combination of glucose and fructose. It's from disaccharide. And a sucrase can break the bond between the 2 molecules of glucose and fructose that can form glucose and fructose. For the detection, we adopt a theory about the special carbon in the molecules of sugar. For sucrose, it has special bond in the red color, red circle. This carbon are called anomeric carbon. You don't need to know what exactly it is because it's very complicated. It's go into organic chemistry. If you don't think you are a fan of organic chemistry, just know that it's a special carbon in the sugar molecules. When it is forming a bond with another sugar, that bond is already inactive and we call it this sugar as the non-reducing sugar. But if this anomeric carbon or the special carbon is free from forming a bond, this will resulting in a more active sugar, as we call it reducing sugar. It is very active. So in our experiment, we will detecting the presence of the products because the sucrose, our substrate, is inactive. It's a non-reducing sugar. But the product is the reducing sugar is very active. It will do another reaction with the Benedict solution. It will form a precipitate. It will form a color that we can observe. So in the case of sucrase, you will detecting the presence of products in order to see the activity of an enzyme. All right, I think this is all for lecture 2, metabolism and enzyme. Um, Here I have some summary for you to read. Everything that you need to know from this lecture. What is metabolism? What is free energy? Exergonic endocytic processes? Activation energy or transition state? How about an enzyme? What an enzyme do? What is a native state, cofactor, and active site? What are they? If you have just, well, I don't know what she talking about, go to these 3 books or any biochemistry book. You can read Leninger Principle of Biochemistry. You can read Biochemistry from Voigt and Voigt. You can read Chemistry and Molecular Biology of Plants from Buchanan. These 3 books are great, but if you have other biochemistry book, I believe they are interchangeable. So this is all for today, and hope you guys have a good day listening to my lecture. And don't forget to do the quizzes that will coming along. Bye.